Thank you so much for your patience. Let's get started today. So welcome everybody to BioRender 101, Improving Communication with Visuals. Just there we go. So what we'll cover today is a brief introduction about BioRender. And then of course, I'm sure what everybody is waiting for is the BioRender demo. Again, just to reiterate, if you do have any questions, please ask them in the Zoom Q&A. And we're also going to run a quick poll just so we can get to know you a little bit better. So just ask a couple quick questions. If you could please answer them to the best of your ability, that would be fantastic. Again, this also helps us just to understand our users at BioRender a little bit better. And then while the poll is still running, I'll continue along with our introduction. So today in the chat box, we have both Cindy and Jerry. Sorry, I forgot to add a picture of Cindy here, but maybe if you two want to unmute yourselves, Jerry first maybe, and you can just tell the attendees a little bit about yourselves.
Yeah, sure. Thanks, Joe, for the intro. So my name is Jerry. I'm working here at BioRender as scientific content lead, and also a little bit in terms of user experience consultation. So working primarily with creating content for all scientists on the platform to accelerate their research and create clear science communication even faster. I think for the webinar today, which Joe will be leading, I'll mostly help out with any questions that anyone may have in the question and answer section of Zoom or in the chat. And also maybe I'll interject from time to time, Joe, if you don't mind, during your actual demo with maybe some additional tips or just maybe some design insights as I see appropriate.
Yeah, absolutely. Yeah, absolutely. Thanks so much, Jerry. I don't know if Cindy has a microphone handy, but maybe if you just want to pop in and say hello.
Hey everyone, thanks Joe. Yeah, as Joe and Jerry said, we'll both be in the Q&A section monitoring any questions you have. So definitely check that out if you have any questions.
Awesome. Thank you so much, Cindy. So a little bit about myself. I'm Joseph Zeppa. I did my PhD in microbiology and immunology at the University of Western Ontario in London, Ontario, Canada. In 2017, I then took my research skills to the University of Pittsburgh, where I did a 3-year postdoctoral associateship under Joanne Flynn, studying tuberculosis in a non-human primate model of infection. So I have quite a bit of academic research background. And just a few months ago, I joined BioRender as a science solutions consultant, as well as a customer success manager. So I'm really looking forward to running the webinar with everybody here today. So our story with BioRender really begins with one of our co-founders, Shiz, who was the lead scientific illustrator at National Geographic for 10 years. And so what Shiz was able to do is she was able to generate these beautiful, scientifically accurate images and provide them essentially to the general public so that they could learn in a, of course, beautiful setting such as this. Now you see here, we've got this amazing image of a shark and the internal anatomy and their workings. And while Shiz was doing this amazing work, what she realized was that not everybody has access to the tools in order to generate such beautiful, scientifically accurate images. And of course, some of the people who need to do this the most, such as researchers, educators, and scientific communicators, didn't have access to the tools to do this, nor did they have the funding in order to get somebody such as herself to make a beautiful image. So out of that idea came the thought, well, what if there was an easy-to-use platform for science communicators to be able to generate their scientific content and get it out there in a very, of course, easy fashion. Another problem that popped up that Shiz wanted to remedy was this idea of confusion when it comes to generating and understanding scientific principles. So what you're looking at here is actually, if you just go into Google, go to images, and search immunological synapse. So this is where a T cell and its T cell receptor recognizes an antigen from an antigen-presenting cell in the major histocompatibility complex molecule. So as you can see here, you have a wide variety of both scientific and artistic prowess at generating these figures. So this one looks like it was maybe made in something like Microsoft Paint. This one may have been created in PowerPoint or Google Slides. Then we have some very beautiful scientific imagery, but of course, even though these are all trying to convey the same scientific concept, they all look very different. So what if we could give, again, scientific educators and researchers the tool to standardize this understanding? So all of these different searched images here, what if they could all look very simple and beautiful and elegant, just like this? And that's exactly what we hope to do here at BioRender, is standardize this scientific imagery so that everyone can, of course, not only create the content, but understand the content. And so what exactly is BioRender? It's an online application tool, which means all you need is a web browser and the internet. And it's a drag and drop process that allows you to quickly and easily create these beautiful scientific images, most importantly, quickly and easily, but also with little to no artistic background. So we, of course, have tried to do a lot of the medical illustration legwork here, so you can just drag and drop these beautiful scientific images onto a nice blank canvas. And of course, then hopefully your concept is much easier to understand than with using something like, again, Microsoft Paint. We also have some amazing integration tools here. One of them that's popped up now a couple of times is our PDB integration. And so in today's demo, I hope to do my best to go over as many of the features that are available to you as possible in BioRender. And so some of the most common use cases for BioRender are figures for publications or graphical abstracts, of course, presentations in your lab meetings, grant applications, research proposals, and the list goes on, as you can see here. But I think what I really like to highlight about BioRender is your imagination is kind of the key. Anything that you can do and anything that you can think of that you might want to use scientific figures for, we highly recommend that you attempt to use BioRender for that, because we guarantee you it'll be easy and quick. And so, of course, our goal here at BioRender, just to reiterate, is to give every scientist, and as well as that, scientific educators, as well as scientific communicators, the tools and practical knowledge to visually communicate their research. So with that, I'm just going to stop the poll here. And thank you so much. We had a very good outcome. And here, I'll actually just share the results with you so everyone can see. So, we've got a very good number of molecular and cell biologists. We always see a nice outreach from our cancer and oncology researchers. And a lot of people have not used BioRender before. So this is actually going to be perfect because we're going to go through right the immediate steps as well, not only for those individuals who have not signed up, but we're just going to show you exactly how easy it is to create your first scientific illustration using BioRender. So I'm going to stop sharing this. Also, welcome back to the individuals who have used BioRender. We love our patrons, and hopefully you can garner something new from this experience as well. So now we're going to move over to a short demonstration. So I'm just going to, exit out of this, go over to here. Hopefully everybody can see my screen now. I am hopefully showing you the BioRender homepage. Just let me know, Jerry or Cindy, if I'm not. But of course,
That's good.
Oh, thank you so much, Jerry. Yeah. So of course, this is just biorender.com. If you go over to that website, you'll see our full web access. And I just want to highlight over here, if you're one of the individuals who has not signed up, you can sign up for free using this link. But for everybody who has signed up, and of course, who are the constant users, they'll know our sign in button. Enter your credentials, and you'll come to our BioRender application homepage. So I'll quickly go over what this shows. So the first thing you'll notice is this big open area to the bottom, which is your My Illustrations area. So essentially what this is a area where all of your generated content goes to. That includes the generation of your own illustrations, as well as if you have used one of our BioRender templates, it as well will populate into this area. So what's really cool here as well is that it's quick and easy to preview, make a copy, as well as share your illustrations. So if you just click this share button, you can send this to anybody who obviously has an email address. And what's really cool about this is that by sharing these, those individuals can then open them up in BioRender themselves. Even if they don't have BioRender, all they have to do is sign up for a free version, and they'll then get access to that illustration that you send them. You can also create and share items quickly and easily with a team. but I'm not going to go over that today. Another cool feature we have at BioRender is this version history. So you'll notice throughout my demonstration that I won't hit save once. You can if you really would like to, but BioRender saves in real time. And what this allows us to do is you can actually go back through the dates that you were accessing your illustration, and you can click on any one of these time points, and you can make a copy of the version of that illustration, or if you were editing a template. And so that just gives you quick and easy access. If you made a change that you weren't a big fan of, you can, again, quickly and easily go back and make a copy of an older rendition of your illustration. If you're anything like me, you also love to organize things. So we have a beautiful, nice and easy folder category. So you can just go my new folder, hit save, and of course it'll populate right along the top, and you can move this anywhere up and down the list you see fit. Then it's as quick and easy as dragging and sliding your illustrations into those folders, and of course they're right there and ready to go. So there are two major aspects of BioRender that set it leagues apart from any other scientific illustration software. One that I'll go into a little bit later is our BioRender templates. So these are pre-curated illustrations that were made by our scientific illustration team, of which Jerry heads. And so essentially, these are scientifically accurate depictions of a number of different topics, and you're able to easily go in, click on these, and edit them. And so actually, the last thing we'll do today is I will show you how easy it is to go in and edit one of these templates to make it your own. But I highly recommend you go into our template library along the top, search any topic that you might be interested in, and take a look and see what we have. Because I guarantee you, you'll find something that you'll love. Another cool feature that has come out of this amazing illustration graphical abstract contest we had earlier this year was that you're also still able to view all of the submissions from that contest. So you can still go back through and view all of these beautiful illustrations. And these are, again, made by BioRender users such as yourself. So these weren't made by us at all. You can see all of the amazing content that was created by our users. And you can even still click into them and view their description, see what awards they won. And if you're interested in the scientific topic, you can, of course, look up the references for those works. Another amazing initiative that was tackled by Jerry was some of these are actually now being converted into templates. So these are user-generated content, so our community gallery, that can now be used by you in BioRender. So if you like any one of these illustrations, you can now go in and edit them yourself. So those are just a couple really cool aspects of our BioRender templates that I wanted to highlight. But what I really wanted to show you today is how you can create your own new illustrations. So for those of you who missed that, to start your own illustration, you just go to the top left-hand corner here and click on blank illustration. You can then name this illustration, my first illustration, and you can note that we actually give you a number of pre-curated illustration sizes. Now, I really wanted to emphasize as well that no matter which one of these you choose, you can, in the future, change the size of this slide. So if you know you're making a journal figure or a figure for a poster or a slide for a Google slide presentation, we do have sizes that kind of pre-fit most generic use cases, but by no means are you pigeonholed into using this specific size. And I'll quickly and easily show you how you can change this in just a minute. So after you've selected your size, you can click Create, and you get transferred to our Canvas view. I'm just going to click this little tab here to give ourselves a little bit more space to see our beautiful canvas. So what we'd like to imagine this as is a blank piece of paper on a table. So everything that you include in this white space will be included in your final illustration, and everything in the outside binding gray space is a workspace for you to upload images or references that you might need to create your scientific illustration. And as long as you include them and keep them only in that gray space, they'll never end up in your final product. But it's just a nice, easy way for you to have an area for you to upload reference images. So along the right-hand side here, we've got a quick and easy toolbar. You can zoom in, zoom out. These ruler guidelines along the top can be toggled on and off. And so as you'll note, we had a 10-inch by 7-inch figure that we selected right off the bat. And so that holds true with our ruler. And you can kind of quickly and easily add some grid lines or guidelines if you want. So if you want 1/2-inch margins, these can be quickly and easily added. So if you want to get rid of these as well, you can just right click and you can either delete them or hide them or lock them. So there's tons of functionality here. If you're used to making figures with graph paper, we also have a grid function where this just automatically adds a number of grid lines that can be made larger or smaller. So I'm going to toggle that off for now, but I just wanted you to be aware of these features. We also have a preview in grayscale. I will come back to this a little bit later once we have some icons on the page, but this is a very handy tool, not only to make sure that your colors on your canvas are working well together, but it also gives you an understanding of potentially what your colorblind audience might see. And so it's always important that before you export one of your illustrations, you take a look at it in grayscale. And this, again, will give you that insight to make sure that everything looks all right, especially from those individuals who can't see different color contrasts. So let me pop this back open. And I guess the last thing I do want to highlight as well is these bottom right-hand corner help and FAQ section. So throughout the demonstration, I will be copying and pasting things very quickly. You can right-click on any one of our icons and function or do a lot of these functions, but I wanted to provide you with this little tab here that shows you all of the common keyboard shortcuts. And as I use them throughout today's demonstration, I'll be calling them out. And that way, you, of course, can get a feeling for how somebody who uses BioRender regularly might quickly and easily copy something, paste something, undo, etc. But this list right here gives you the full gamut of our keyboard shortcuts. We also have a helpful FAQ help guide link. So if you just click that, it'll take you to our FAQ page. So if you feel like trying to tackle a problem on your own, this is what I recommend that you go and look for. And we also have a live chat box, which by clicking on that, you can talk to one of our amazing customer experience representatives. And these individuals are live on the line Monday to Friday, 9 to 6 p.m., and that's Eastern time. So just to be sure, if you're in a different time zone, the time lap might not be perfect, but we do have people that are on the clock to help you as well within those time frames. So let's get to making our first illustration. So if you recall earlier, I mentioned kind of there was two things that really set BioRender apart from any other scientific illustration software out there. Number one being those pre-curated templates. And these are in no specific order either. But the next thing for sure are our icons. And so our icons are these amazing vector-based images. Let's go through and pull out a mouse, because I know a lot of people like to use, scientists have animal models here. So as you can see, by pulling out one of our icons, you can scale this to essentially any size, and we don't lose any resolution or contrast. It's just a beautiful, seamless transition from larger to tinier. You can also go up to the top and quickly and easily change these icon colors. So again, if you're anything like me and you have some rodent research experience, you have your bulb C mice, your C57 black 6 mice, any sort of wild type mice sometimes come in these lighter tan colors. As well as if maybe you want to indicate your mouse expresses a GFP or an RFP protein, we also have common case uses for that. So that's how quick and easy it is to drag out an icon as well as change the color. What I hope you've noticed in this icon search box is the way that I got into that was just by clicking through these easy to navigate tabs. What I also want you to note is you can actually just search for any icon that you may want. So we have a mouse out there. Let's type mouse again. And you can see just by typing in mouse, we have these beautiful images, again, of our mouse as well as mouse anatomy that pop up. So let's pull out this icon as well, because as you'll notice, this mouse comes in this beautiful blue bounding box, but this one has a purple bounding box. So the difference between these two entities is that the purple bounding boxes just give you a little bit more editability. So as you can see right away, double click to edit this group. Let's do that. And as soon as you do that, you'll notice the rest of the screen grays out and only our purple bounded image becomes colorized. And you can actually notice now that each one of these aspects of this mouse can be edited. So we've got the entire mouse in the background, so you can still change your color. But maybe you're only interested on the internal thoracic avenues of this animal, I guess the intestines, and you're not a big a fan of the esophagus and the palate. So you can quickly click on those, get rid of them, and then double click out, and this is still a grouped icon, but now you've gotten rid of the aspects that you're not as big a fan of. And so I think you can go through, and I'll just use some other common species that we might be used to. So here's a, classic C. elegans, one of the other most common used lab animals. I worked on non-human primates for a couple of years, so I was really excited to see that they had the species that I worked on, a rhesus macaque. Again, these are just quick and easy to drag out. One of my favorite icons is our fruit fly. So you'll see here we have these beautiful, different colored fruit fly renditions, kind of indicating different genetic mutants. But what I hope you'll also notice is we have a couple of these really cool grouped animals here, or sorry, grouped icons here. And if we just kind of scroll over to it. So I'm on a trackpad actually right now. So I'm just panning by using my 2 fingers on the trackpad. I'm also using a Macintosh as well. So I will be calling out Macintosh-based shortcuts. But again, if you're using a PC, the keyboard shortcuts that you observe here will 100% show up for your keyboard shortcuts using a PC. So back to our awesome fruit fly here. Essentially, every aspect of this is editable. You can change and move around the wings. You can move around its thoracic cavity. And you can also, of course, change the color of its tail or of its head. And then clicking out, this whole entity is still grouped together. So in this kind of, just pulling out icons, looking at how awesome they are no matter how you scale them, another really cool feature that I like to show is our zoom feature. So essentially, say we're looking at mouse anatomy and we're really interested in that. And we can zoom in here in BioRender and we can scroll over and we can get a really good understanding of what's going on here. If we wanted to demonstrate this from more of an aspect of zooming in as if you're using a magnifying glass, you can just type in zoom into the search bar and you can drag out any one of these zoom icons. And you can see that we can kind of position this so as if the smaller circle is looking over this part of the mouse. So I'm right now just holding the Option key, clicking and dragging, and hopefully you can see how that easily and quickly copied this mouse. So I want to indicate that I'm zooming in on this part of the animal. So let me just blow up this aspect of the animal using these side guiding boxes. So now we have, of course, a rather hilariously large mouse beside our smaller mouse, but this is a great time to introduce our crop function. So of course, when you click crop, you have your classic square-based functionality. And of course, this is good for cropping, anything from our more square-based icons or, whatever your imagination can conceive. But what I really wanted to highlight here was our circle crop. So what that does is, of course, just generate a circle that you can then highlight over an area of interest. So for us, it's going to be the internals of this mouse. And then after I'm done cropping, I click out. And as you can see, it almost looks like we're zooming in again on that portion of the mouse. So again, just a couple other cool images I'd like to pull up. We've really thought about our human and human anatomy. So we have a number of different male, female, nondescript humans, different ages, adolescent, baby. We also have kind of a similar human anatomy that you can pull out. And with, you could probably do the exact same zoom functionality with this. As well as this is actually a pretty perfect time to highlight grouping. So much like you can pull out these grouped icons that are purple bounded, if you're interested, you could actually create your own. So say this is already a grouped icon, but say we really like adding this zoom feature to this mouse. We can highlight these two, and you'll notice that we not only highlight the zoom and our original mouse, but you can go up along the top to group, and now this is one grouped entity. So you can quickly and easily group and ungroup items. Another cool feature to kind of bring this home is this favorite functionality. So say you're consistently using this zoom in of the internal anatomies of a mouse, and you don't want to have to keep recreating this or copying it and pasting it between different illustrations. We have this amazing feature where you can either right click and hit favorite or hit favorite along the top toolbar. And you'll notice along the left-hand side, a number 1 is added. And you'll now see that this image that we just created, let me delete it here, is now added to our favorite section. And you can now quick and easily drag this image out whenever you want to use it in the future. And you'll see here I already have populated a couple more: infectious disease, virology, immunology, maybe a little bit of cancer. So you can kind of curate this list on your own, and it's kind of like your own BioRender icon playlist. So whatever you find yourself using most often, we highly recommend that you favorite it, because this will only save you time. Also, if you happen to have created one of those teams that I mentioned earlier, you can also share these different favorited icon or grouped icons with that team. So say you want your whole team to use this icon to denote imaging the internals of a murine model. You just click on the share with your team. You'll notice, of course, I'm on the BioRender team, but this will say whatever you happen to have called your team. Make sure that this box is checked, hit update. And now when you go over to team, you'll see that these are now shared with anybody who I have on my team. So anybody who's on my BioRender team can also use these icons. And if you don't want them to see it anymore, hit that team button again, unclick the check mark, update, and now it's gone. And now it's only available to you in your favorites. So I hope that this has done a good job of showing you exactly how quick and easy it is just to pull some icons out of our amazing icon library, their high resolution and contrast, as well as how quick and easy you can edit these grouped icons. But let's start a blank illustration to go into something that's a little bit more looking like a figure. So say I want to make a signaling cascade, a membrane signaling cascade. So let me just type in membrane here. And you'll see that we have these beautiful brush icons, which I'll show you a little bit in a second. There's a few of these that are free access, but the majority of our brushes are premium access. So I don't like to overuse them in this demonstration. I want to show you exactly what you can use as a free user, but we will highlight them a little bit because they are a pretty miraculous piece of technology. But to start, let's just pull out this membrane. And you'll see it's relatively small here, but as I mentioned before, we can kind of expand this as big as we want, and it will look pretty good. So you'll notice that I'm doing this, and I love how we have these beautiful lines that denote exactly when it's in the center. Again, I'm purely an academic background, so my artistic skills are pretty limited. So all of these little features I absolutely love in BioRender. So again, quickly and easily pull that out. You can skew it to make it bigger. And again, you can change the color if you want. I'm just going to keep it this gray-y blue color for now. So another thing that I love about BioRender is this lock object. So you'll note that I just clicked locked object. When I click on my membrane, it now says that it's locked. And what that means is I am pushing down on my trackpad and trying to move this object. I can no longer do that. So in the past, if you ever created an image and you started layering other objects over this, so say we added something like a TLR, we can quickly and easily pick this up and drop it on. If I was trying to now orient this in some way, but accidentally missed it and grabbed the membrane, I may have accidentally moved it and of course been a little bit frustrated that I had done that. So I'm just using Command Z to undo that action. But now with this lock feature, I do not have any worry about ever doing that again. So we just beautifully pulled out this toll-like receptor, orient it in the membrane, and maybe we're trying to show a sequence of this TLR being internalized. So we've got this nice toll-like receptor. I'm going to, again, use that handy holding down option and just pulling it over to replicate it. And then maybe this is being internalized into a vesicle. So I actually will use the brushes for this vesicle demonstration just so I can show you how functional it is. But maybe right off the bat, I'll actually just show you something cool like our DNA. So all of our brushes are denoted by this little paintbrush in the bottom left-hand corner. And I'm sure many of you in any sort of the molecular sciences, as we saw, there is a fair number of you in the room. If you're ever used to dealing with or drawing DNA, it's a bit of a pain. You have to copy and paste all of these base pairs together. And if you want it to be longer, you either have to kind of grab it from the side and enlarge it. And it never really fits into the exact space you want. So hopefully at BioRender, we have fixed that for you by creating these beautiful brushes. So you'll notice a couple blue, and then if I zoom in a bit here for you, this white node in the middle. These blue nodes on the end actually allow you to grow the DNA in real time. And as you grow it out, you'll see not only does it not skew, but it actually recalculates and now creates a seamless nucleic acid strand without having to copy and paste anything. So these blue nodes are, as I'll show you in a second, very much like how our arrows work. They give you the free-forming functionality to just manipulate the strand in any direction that you want. And after you let go of clicking the blue, it sticks into that orientation that you've manipulated it. These white nodes simply just add new blue nodes. So if you want to add multiple bends to your DNA strand, you'll notice here that it actually looks kind of funky when you're doing it, and it looks like it's almost broken. As soon as you let go, it recalculates and adds the strands back together in that seamless fashion. So hopefully, this is a good demonstration of just how quick and easy it is to use our brushes, how to grow them. And we actually have brushes, again, for numerous different things: phospholipid bilayers, for bacterial membranes, as well as for cell lines. So this is kind of cool. You can, create your own columnar cell brush, enterocytes, epithelial cells, etc. So, and if you ever add a blue node that you're not a big fan of, you can just quickly and easily double click on it and it goes away. The full brushes options are right here. And you can, of course, adjust the size. Doesn't make sense because DNA is obviously a mirror image of itself going back and forth. But you are able to flip the strands if you're interested. This makes more sense once you get into some of our cell brushes. Cool. So that was kind of my intro to the membrane brushes. But as I mentioned before, I'm just using this now to draw a nice vesicle. So you'll notice the colors don't necessarily match. So I'm just going to go in and make that quick change. Perfect. And then I'm also going to copy. Oops, let's bring this to front. Send to front. So you can also see that when you go over something that does have a shortcut, it tells you right beside it. So if you ever wanted to get really handy with BioRender, you can just memorize all of these shortcuts and save yourself an amazing amount of time. So here we go. We kind of have this process where our TLR is being internalized, but obviously it would be nicer if we had some way of depicting that. So of course, I think the most natural way of showing this would be by using arrows. So we here at BioRender have gone into agonizing detail with our arrows. You'll see here that these top 5 different types of arrows look very unique and very editable, and I guarantee you they are, but you're not really restricted to any one type of arrow once you create one. So of course, the most logical step here is to maybe draw an arrow like this and show that this TLR is going into this vesicle inside the cell. But say I actually wanted to indicate that this wasn't happening, that this was actually being inhibited. I wouldn't have to delete this arrow and then draw a new inhibitor arrow. You can go along the top here, and as soon as you click on one of our arrows, you'll notice this beautiful new toolbar pops up. And you can do a number of things. You can adjust the line color. And of course, we have this cool color picker. So if you have, picked an icon that has a color that you love, or if you've imported an image that you have a color on there that you like, you can, of course, just go like that, find your color, and kind of steal it. I'm going to keep it as black for now, but I wanted to let you know that you can change it to any color under the sun, especially if you have a hex or an RGB code. You can adjust the line size, so maybe we want to make it a bit thicker. You can add dashes or subtract dashes, add a line fade if you want to denote maybe a movement action, as well as you can quickly and easily swap the head and tail back and forth. But kind of back to my thought, maybe I want to make this an inhibitor. Just by clicking on the head or the tail, this useful menu pops up, and you can, in real time, switch the type of arrow that you have. And of course, much like our brushes, you have these blue nodes that allow you to add flexibility and customization. and these white nodes that, again, just add new blue nodes. So I'm going to go back to my arrow, remove the line fade. Perfect. I am going to then copy this arrow as well. Maybe I want to orient it a little bit. Get rid of this. Go back up, make it a straight line. No, let's add a bend back in. Looks a little bit nicer. So another thing that we've really thought about here at BioRender is quickly and easily making not only text boxes, but text in shapes. So just by going to Insert Shape, you can see we have your classic stars, squares, circles, triangles, another family of arrows that don't quite have as much manipulatability, but they still are, very easy to click and drag out. But what I wanted to show you here is labeling steps using these text and shapes. So you can just quickly and easily draw out this text shape that says 1, and then maybe you want to insert a text box that says something like step, or phase 1. Cool. Whoops, not phase 12. A little bit too many. And of course, as soon as you go like that, you can see there is a red line that allows me to align different objects together. So maybe I want to say this was phase 1, the internalization, and then I'm just holding Shift to select multiple entities. And then maybe this here is our second phase. So we're kind of missing a little bit of, entities that are normally inside of a cell here. So maybe we can go up here to icons and go search, and maybe we want a nucleus. And so you can see here we got a nuclear membrane brush. We have a couple different prefabulated membranes that are filled, but I'm just going to use this, nuclear membrane that's porous. Click and drag that out. And I'm going to also give us a little bit more room here. And again, cells generally have an endoplasmic reticulum. So if I even just start typing something, and maybe you're self-conscious like me and you don't want to make a mistake in front of all of the attendees, you can kind of just stop a little bit premature, and you'll see that most likely BioRender has you covered. And you can see here that it's kind of quick and easy to drag and drop out your own endoplasmic reticulum. Maybe we want to make this a little bit bigger. There we go. And you'll see here that it's kind of cool that we can add this in. So I'll give you an example of some of the prefab DNA that we have. We have all of the brushes that I mentioned before. We also kind of have these pre-made strands that give you, a nice amount to work with if you don't feel like working with the brushes right away. So, you can just quickly and easily throw that in. Actually, I'm not a big fan of that one. Let's grab this one. There we go. And then another pretty popular feature for using our arrows are indicating that, maybe a gene is being transcribed. So you can kind of quickly and easily draw this out. And maybe we'll add kind of a nondescript protein here as well. Sure. Add that one in, make it a bit smaller. And I'm not a big fan of that green color, so maybe I'll make it a darker blue. Cool. So then you can see how you can quickly and easily indicate that maybe a specific gene is being transcribed there, and that is all a sequence of this TLR being internalized. So maybe we'd like to add, another arrow, and this is our phase 3. Perfect. So again, kind of with this arrow functionality, just looking at any one of our text icons here, you can see that you have this fairly customizable toolbar along the top. You can change the style of text, the size of text. You can bold, you can italicize, underline. Much like our lines, you can use the color picker to steal a color. If you have a specific color, maybe your school's colors or your company's colors, or just a color that you're rather fond of, not only do we have this beautiful palette of colors that work well together, it's really cool. You can kind of see the color changing in real time. So you can kind of see which one you might like best. But you can, of course, also enter your hex or RGB code. I also forgot that I mentioned earlier how easy it was to change the canvas size. So maybe I realized, oh, I'm cutting it a little bit close. I want to give myself a little bit more buffer room here. It's as quick and easy as clicking canvas size. We see the predetermined size that I chose initially, the 10 by 7. This is, of course, in inches, but if you're like me and you prefer the metric system, we have centimeters there for you. Not only can you edit the canvas size by entering your own numbers, much like our crop functionality, you can actually just click and drag the corners out as well to give you a little bit more room. And then that way you can just apply it and you get the idea of just how easy it is to kind of adjust your canvas size. So kind of going down the left-hand side here, we've searched icons. I'll quickly highlight this. All of those templates that I will quickly go into showing you how you can edit them in just a second here. But you can pull in any one of those templates from our template library, and you can actually integrate them into any one of the illustrations you're building. So of course you can search them, but for the interest of time, I'll just click this SARS-CoV-2 one. You can see that this is one of our user-generated templates. So we have the individual here who created it. And so you can either scrap the illustration that you're making and replace the template, or you can just add it to illustration. And this will be imported into your canvas view as a grouped icon. So you'll see this one's relatively busy, so maybe it's not the best example, but we can kind of scale it down here. And you can see that maybe this had something to do with what we were drawing, and you can just throw it in the top right-hand corner there. And so, hopefully you can see how quickly and easy it is to integrate our templates into your illustrations. Uploads are another very important feature as well. So say you were creating this image and you were part of a company or a school and it was going into intellectual property or in a presentation and you really wanted people to know that it was yours. You can essentially upload any TIFF, JPEG, or even vectorized files, SVGs, and you can quickly and easily, again, bring those into your own illustration. So I've got a couple pre-fabulated here, so I'm just going to bring in this BioRender image. So again, you'll notice here that when you hover over this item, this also has team share capabilities. So maybe we want to throw this into the top right-hand corner. That way it's kind of out of the way, it doesn't take up too much space. And we really just want to indicate that this is BioRender image, BioRender property, what have you. So another really cool thing that I want to highlight is this opacity knockback. So some people might be thinking, why would you want to, fade out an image you just brought in? But I've noticed that you can actually relatively easily, I always find 40 seems to be this magic knockback percentage, kind of gives it that watermark appearance. And so I think this looks really cool. We demonstrate this a lot with different companies we work with, just to import in their logo, knock back to 40% opacity, and it adds that kind of watermark functionality. So people doesn't take away from the science, but it definitely adds something to the image. I guess the last feature I haven't quite highlighted here just yet is our integration with PDB. So for those of you who aren't aware, the PDB, or the Protein Data Bank, is an online repository of crystal structures that have been published into things like Google Scholar, PubMed, etc. So you can just honestly go on and Google Protein Data Bank or PDB, and it'll pop up. You can then go into that database and search for your protein of interest. And upon doing so, you'll notice that it comes up with this 4-character alphanumeric code. So what you would do with that code is you would go into our PDB integration, go create protein. Again, if you know that protein ID, you can add it in here. I'll just maybe use one of the ones that we have down here at the bottom for the purposes of this demonstration. And your protein would then load with not only that code, but the name of what it was. So, if this so happened to be the one that I had searched and chosen, it is the structure of the human glucagon class BG protein coupled receptor. And hopefully what you know right away is that you can now manipulate this structure in real time. And you can, of course, zoom in, zoom out. A whole list of our controls are right here. But moving forward, you can switch over to a cartoon view if that's what you like. Keep it as our fill, look and licorice structure, etc. So again, I highly recommend you come in and play with some of these features. You can, of course, then change the color. based on things like sequence, hydrophobicity, or if you just want a uniform color, you can do that as well. You can change the border style. I'm a big fan of doing a thick border with a toon style shading. I think it looks relatively sharp. Then once you have this oriented exactly how you want it, you can save it. And it now not only pops up on my screen here, but it also is curated into my PDB library. So you can see kind of all of the other PDB molecules I've played around with over the last few months. And then, of course, what I really like here is kind of going back to the beginning, that zoom functionality. Maybe we want to demonstrate that, this part of the TLR has that structure. Let me actually just fix this a little bit, bring it up, apply. There we go. Gives ourselves a little bit more space. So you can kind of see how cool and easy it is to then integrate a protein data bank structure into our illustration. So I think I'm going to show you now what happens with our export modal. So quickly, you can always preview throughout the demonstration. You'll notice that everything that's in that gray area doesn't show up, and everything that is within our white canvas view is there. And then, of course, another feature that I think is important is, of course, our export modal. So as soon as you hit the export in the top right, you can see the dimensions that we had. Of course, you can change this even up to this point. We have 3 different export types, PNG, which is recommended, JPEG, and PDF. The reason why PNG is recommended is because we have this transparent background modal as well. So all of this white space now in my export will actually become translucent, or I guess transparent. And thus, if you have another background that you really hope to show through in a presentation, then that's a very handy feature as well to have. And then, of course, we have different resolutions as well. Now, I think for all of the resolutions, you need our paid premium version, but you do have 72 DPI that's available for the free version, and this does look relatively good as well, but I'll just stick to 150 because we're going to import this into our presentation at the end. So I'll export that. I'm using Google Chrome, so it actually just pops right into the bottom of my browser here. And of course, we always prefer that you cite BioRender whenever you use it, especially when it comes to publications. And so we ask that you just use this phrase here, created with biorender.com, and you can now quickly and easily copy this to your keyboard shortcut just by hitting copy, and then you can paste that in the relevant location, which is usually, we recommend in the acknowledgements or in the figure legend if this is a publication. So I said we've got about 9 minutes left. So I think the last thing I did mention that I would do is indicate how quickly and easy it is to edit one of our templates to make it your own. So I'm going to go back to gallery. So another thing I really hope you noticed there is I didn't hit save once, and both of my 2 illustrations are now right here in my illustration view, complete with all of the different version histories from the last, 15 minutes or so from creating this image. So hopefully, again, that gives you a little bit of peace of mind. If you're creating an illustration and your web browser crashes or your internet crashes, you never have to worry because you'll never be, more than a minute off from having your image that was there when maybe your web browser crashed. So let's go to template. I'm going to quickly show you how you could edit something like a protocol. So let me just start typing in mass spec. And as soon as I type in mass, of course, mass spec pops up. So use this template. And here we go. Our peptide mass spec pops up. And it's kind of a really nice, beautiful layout already. But maybe I'm not working on brain. Maybe I'm working on liver. So again, we can just quickly and easily throw in liver. And then maybe also to demonstrate the upload function, not only can we upload things like logos, but you can upload data and histology slides. Let me pull this histology slide. Let me go like that. And we can kind of show that maybe this is just indicating this is what livers look like histologically. And, now we want to sample this. So maybe we want to show as well some cells in our Petri dish. So I'm just going to pull out this grouped icon of cells. And I'm going to go like this and throw them into our Petri dish here. And I, 99.9% of the time would never recommend you skew any icon vertically or horizontally alone. I always think you should skew things using the boxes on the corner because that will scale things in real time. But the one exception I found to this rule is with these Petri dishes, you can kind of skew them like that and they almost look like they're sitting on the Petri dish. So there's your one exception to the rule. But again, it's a very rare occurrence when you should skew things using these sidebars. And also, maybe I'm a big fan of purple. Maybe I like this purple on red, and I think it looks pretty sharp, especially because my histology H&E stain also looks purple. So this is where this preview in grayscale that I highlighted in the beginning actually is so important. So let's zoom in a little bit here. And maybe I think that this looks fantastic. And yeah, purple on red, you can't really see a problem with that from the perspective of color, but it does look a little bit off to me. So if you throw anything into grayscale, you'll notice how hard it is to see these cells on this agar background. And so again, I always recommend before you do anything in your images that you check it in grayscale because you'll notice these differences in contrast that you can't really observe when you are looking just purely at the color image. So while noticing this, let's go back to that original orange color we had, and wow, that contrast looks way better than what it did previously. And looking back, color-wise, it actually looks pretty good too. And so I'm going to stick with this orange. So that's where this preview in grayscale really comes in handy, not only to help you check your color choices, but also now anybody who's colorblind who comes into this image will be able to see those cells, as opposed to before, they may have actually lost those cells into the background by using this purple color. And so I think the last thing that I'll do just to kind of make this image my own is maybe I'll change the data analysis. I've got this is lung necropsy, so maybe it's not relevant, but this is actual work from my postdoctoral position. And then maybe I want a different mass spec machine as well. And so you'll see we have a number of different mass spec machines. Maybe I like the liquid chromatography mass spec instead. So again, you can see how quickly and easily you can kind of make this image your own. And maybe you don't need, you know it's mass spec. Maybe you don't want the label at the top. So let's just shrink it down a little bit to fit our size constraints. Cool. So hopefully this has been beneficial just to show you how quick and easy it is to edit one of our templates, add your own data, change out a different instrument that you have. I think those are all very important features, as well as that preview in grayscale highlight. So let me wander back. Oh,
Hi,
hey,
Joe.
Jerry.
Just wanted to add one very last point, if you could jump back to the template you were working on.
Absolutely.
Yeah. So when you were working with the swapping out the mass spec machinery, one thing I think it's also helpful to bring up is that if you do find that we do not have a certain graphical icon of a machine or really of any topic you're working on. So an icon, again, is something that's kind of the foundational visual unit of whatever research you're doing. That we do have a request a custom icon option at the very bottom. And this is a feature we have ever since the early days of BioRender, and it's really been a great collaboration between us as a company and you as the scientist community to collaborate and really organically grow the icon library so that it's more comprehensive for everyone. So every time a user requests a custom icon, we follow through, that is added to the general library of icons, and all other scientists, especially the ones who are in the same field as you, will be able to find that icon and now going forward use it.
Yeah, thank you so much, Jerry. It's a fantastic point, and thank you so much for catching that. I always do like to highlight that. So, and as you can see here, kind of the ability to request an icon is simple. We just ask that you give it, a rough name, highlight some key features, provide a description, and you'll see it says three different pages. Of course, these are even less, I think, than the next pages are even less than this information. And we do ask that you attach an image. You can even pull one from your own lab that you snap on your smartphone, or even upload from Google. Just give us some idea of exactly what you're looking for, and that will help our medical illustrators create the most accurate icon possible. So thank you again so much for bringing that up, Jerry.
No worries at all. And I think as you swap back to the slides, there have been a couple of questions from our attendees today about like usage rights, whether they're a free user or a paid user in BioRender, where they can use it for. So I think that might be a good segue.
Perfect. Yeah, so I'll hop back over into our presentation. The last thing I wanted to show you is just how quickly and easy it is to insert that image that I exported earlier. So we're just using Google Slides, insert, upload from computer, going to downloads. I, of course, didn't name anything, but there you go. So now it's in our presentation, looks great. There's that transparent background I talked about before. You'll note that the white space shows through. So of course, bringing it back to what Jerry mentioned here, so with publication rights, of course, our free version is available to everybody. And of course, any image that you create using the free version can be used in things like your thesis if you're a graduate student. It can be used in oral and poster presentations. It can be even used in different social media content that you decide to create as well. Really, once you start upgrading into our individual lab and institutional licenses, this is where you get the access and the ability to, to publish in journals and publish them, publish your manuscript. So you do need a paid version if you do want to include a BioRender created image or template in a journal publication. And so you can find all of our pricing options for both academia and industry at biorender.com/pricing. And that will just direct you over here to our pricing page on the main biorender.com home webpage. And of course, we always like to highlight the premium features. Of course, you get access to our bio brushes. You get access to infinite storage space on BioRender, as well as you get request a custom icon. So if you're a free user, we always accept your icon requests, and we usually like to bring them all together and see which ones are most popular as we create them, because of course we want to satisfy the most users as possible. But as a paid user, you actually get guaranteed icons being created for you. So if you absolutely need an icon to be created, the paid route may be what is best for you then. And I noticed that we're right at our 1 o'clock time point, so I don't want to belabor the point. So if there's any other outstanding questions, I'm sure myself, Jerry, and Cindy would be happy to answer them. But of course, I also want to say thank you so much for joining us. It's been an absolute pleasure giving you this demonstration. BioRender.com is the website. And if you have any questions at all, support@biorender.com is the email that you can shuttle those questions to. Any last minute questions, Jerry?
Just something earlier. For those of you who would like to share this webinar with a colleague or peer who might have missed it today, we'll, of course, be sending out the webinar recording in the coming days.
Fantastic point. Awesome. Thank you so much, Jerry, and thank you, everyone. With that, I think I will end it there. And of course, I'll leave this slide up just for a couple more minutes in case there's other questions in the chat box. But thank you very much for your attention. And we look forward to seeing all the beautiful things you're going to create on BioRender. Have a fantastic rest of your day.
Bye, everyone.